Okay, good morning. Today you are studying about the microbial genetics, the next topic of the biotechnology. So, in microbials or microbes, viruses, bacteria, one process is called as horizontal gene transfer, that is HGT. It is a process of prokaryotic cells, especially in the prokaryotic cell, it can acquire gene from other microbes of the same generation. So the gene transfer from one prokaryotic cell to another prokaryotic cell through a horizontal gene transfer process. The technology is discovered in Japan in 1959. There are three types of the HGT are there. One is transformation, one is transduction, and one is the conjugation. Transduction and conjugation both are in the bacterial cells. So we will study one by one. So how the genes are transferred from the one microbes to one cell to another cell in the microbe. So many methods are there. Apart from these three methods, transformation, conjugation, transduction, these methods are told. Then these plasmid methods are there and transposons are there. So one by one we will study. First one is the transformation. So transformation is the genetic alteration of a cell resulting from direct uptake and incorporation of the exogenous genetic material from its surrounding through the cell membrane. So whatever the surrounding cell membrane, surrounding, it will transformation, the genetic material or genetic messages will be taken into directly to the cells. So you can see here in recombinant plasmid, in recombinant DNA technology, once the recombinant plasmid is attached or once the gene alteration is done, in this picture you can see here. So it can be directly inserted to the bacterial E. coli host cell. This is the bacterial E. coli host cell, and this recombinant plasmid can be directly inserted in this transformed cell. So the transformation into this cell is taken place. So this whole cell is called as now as the transformed cell. So transformation is the genetic alteration of a cell. This is the cell E. coli, resulting in the direct uptake of the exogenous genetic material. Exogenous means outside the cell, whatever the genetic material is there from surroundings of the cell, it will take through the outer layer of the bacterial cell, that is the cell membrane. Second method is the transduction. So transduction is the process by which a virus transfers genetic material from one bacterium to other. So two bacteria are involved here. Transduction means the virus, inside the virus, whichever is there, that is passed from one bacterium to another. Virus is called bacteriophages are able to infect the bacterial cells, use them. Usually in this, bacteriophages are the used viruses to make the host. So this is the transferred gene. This is the bacteriophage virus. So from this bacteria, it is transferring to this bacteria. So this is the transformation, you can see here, and this is the transduction. These are the difference between the donor cell and the receptor cell, acceptor cell. This is donor and this is the transformation, direct intake. But transduction is only the movement of the viruses from this transferred bacteria to this bacteria, one bacteria to another bacteria. Transduction is the process. Only viruses can be transferring through that viruses the genetic material is transferred. In the transformation, directly whatever the genetic material is taken place. In the transduction, this transformed gene, you can see here, it is transferring from one bacteria to another bacteria with the help of the viruses, that is the bacteriophages. Next is the conjugation. In the conjugation process, by which one bacterium transfers genetic material to another through the direct contact, same as the transformation. During conjugation, the one bacterium serves as the donor of the genetic material and one as the acceptor. So these are you can see the differences of the transformation, transduction and the conjugation. So you can ask the question in here. Conjugation also direct genetic transfer, and transformation also direct genetic transfer. What is the difference? So difference is, in this transformation, the cell lysis is there. Here you can see the cell is dying with the lysis process. Again, recipient cell, whatever the genetic material is there, recipient cell, it will attach to the recipient cell. But whereas in the conjugation, what happens? The donor cell and the recipient cell connects through the cell membrane and direct transfer will take place. This is the conjugation. In the transformation, cell lysis is there. In the conjugation, cell lysis is not there. In the transduction, I told donor cell and the recipient cell. In this, the transgenic material or transfer genetic material through transfer through the cell lysis with the viruses. So this is the transduction. So these are the three differences of the complete genetic, microbial genetics or gene transfer from one bacteria or one prokaryotic cell to another prokaryotic cell. Next transfer is the plasmid. So the plasmid is a small extracellular chromosomal DNA molecule within a cell that is physically separated from the chromosomal DNA and can replicate independently. So plasmid is a material, it is an extra chromosomal DNA within the cell which is separated from the chromosomal DNA, which can replicate independently. These are usually mostly circular, double helix, double stranded DNA molecules. These are present in the archaea of the, and in also eukaryotic organism also these will be present. So you can see here, if it is a bacteria, E. coli bacteria, this is the plasmid surrounding circular. This is the chromosomal chromatin layer. So you can see here, this is the plasmid map. So whenever recombinant DNA technology is used, one portion of the plasmid is taken place. Again, it is reinserted. So these plasmids can also use as a insertion or for the transformation of the genetic materials. Last is the transposons. Transposons or transposable elements. These are defined as a DNA sequences that are able to move from one location to another in the genome of the eukaryotic cell. They can be moved. Transposable elements have been identified in all organisms. So these are present in prokaryotes, eukaryotic, and also in the higher proportion of the specific genomes. Usually fall into two categories. So those that transpose directly as a DNA, those transpose via an RNA intermediate prescribed as a by an RNA intermediate. So directly to the DNA transpose, and through the RNA intermediate they can with the RNA polymerase enzyme, again it is converted to double stranded. So this is DNA by reverse transcriptase process. This is also one type of the transposable element. So in the transposons, two classification are there. One is the retrotransposons and DNA transposons. So in retro, I told already, it is RNA copy and paste. In DNA, direct cut and paste method are there. So what are transposons and retrotransposons? Mobile elements that transpose through a DNA intermediate are generally referred to as a transposons. So through the DNA, if they are transferring directly, that are called as a transposons. Retro means via RNA, again it is a reverse transcription process. So that is called as a retro transposons. Usually these retrotransposons is seen in the retroviruses like human immunovirus, HIV. These retroviruses show the retrotransposons processes. This is the transposon process, insertion sequence of the transposon. So this is the donor DNA. This is in retro. This is donor DNA. You can see here. These are the flank links. So these pieces are cut into pieces. So directly the insertion to the another DNA here. This is direct transposons with the DNA. This is through RNA. So donor DNA attached to the RNA. Reverse transcriptase, again RNA intermediate, again it is changed DNA intermediate. Then it is attached to the targeted DNA. So this is the retrotransposons process and this is the direct transposons process. So this is about the microbial genetics of the PCI syllabus. So 5 types are there majorly what you have studied. One is the transformation, conjugation, transduction, plasmids and the transposons. These are the 5 methods of gene transfer in the microbes.